Hi, I'm Brian Carson, Programme Manager for FHI's Healthy Ageing and Performance Nutrition Programme here at the University of Limerick. We understand that muscle and its function are important for our health into older adulthood and for performance in athlete populations. We know that both exercise and nutrition can impact muscle function independently, but more important is the synergy between the two. In our work with FHI, we are interested in the bioactive properties of dairy-based proteins, i.e. something in these proteins which can influence physiological processes. We use cell and human models to establish the mechanism through which these bioactives confer their benefit. In the first instance, we need to identify proteins which have bioactivity. To do this, we use an ex vivo in vitro approach, whereby a person drinks a milk protein. We then take a number of blood samples from them and then apply this blood to muscle cells which we have grown in culture. Following this, we measure these muscle cells to establish if they adapt their rate of synthesis of new protein within the muscle and subsequently grow. Once we have identified proteins with the greatest bioactivity, we proceed to human trials to establish if these bioactive hits translate into the human model.
Before we conduct human studies investigating the bioactive effect of proteins, we must ensure that we have complete control of participants' diet, so we know that any positive effect is down to the protein being tested. Participants are asked to record their habitual 7-day food and fluid intake and to weight all ingredients of their meals and beverages. In addition, we measure their body weight, height, and total body composition, for example, how much fat and lean tissue each participant has. We design the meal plan to the specific participant based on nutritional recommendation. Taking into consideration their habitual food choices and intolerances, the amount of energy and macronutrients provided within the diet are individualized. To make this possible, we collaborate with a local catering company. They prepare meals and beverages based on recipes and information about ingredient quantities for each participant provided by us. We also prescribe the timing of intake of each of the meals provided. All of these components allow us to minimize the variety of food quality, amounts, number of eating occasions, and time of meals consumed. This control allows us to establish the bioactivities of proteins in humans.
Our first human model examines the ability of bioactives to improve the recovery of muscle strength and function during a resistance exercise program. To do this, we have groups of subjects complete a resistance exercise program, and different proteins are provided to each group during training. Following a resistance exercise session, we measure the athlete's muscle function, take samples of blood, saliva, and muscle, and this allows us to assess whether or not these proteins have a beneficial effect on muscle adaptation and performance.
In our second human model, we are investigating the effect milk-derived proteins may have on glucose uptake in muscle. In the exercise physiology lab, we are performing exercise testing on cycloergometers. Prolonged exercise is known to reduce muscle glycogen stores until glycogen limits the ability to continue cycling. Glycogen stores are replenished during recovery, and this process is augmented by high carbohydrate and protein intake. We in FHI are assessing the effect of milk-derived proteins on muscle glycogen resynthesis during the recovery period and how this affects the athlete's future performance. Ideally, our research will lead to the development of new performance nutrition products designed to optimize recovery from heavy exercise.
Finally, we're interested in keeping healthy people healthy, especially our ageing population. Muscle and bone quality are important for healthy, active, independent daily living. Protein consumption has been shown to be an important factor in the prevention of sarcopenia and osteoporosis, both of which we measure using a DEXA scanner. FHI have previously shown that simple daily ingestion of milk proteins can have an effect in not only slowing the decline in muscle mass, but actually increasing it. We are investigating the effects of bioactives on muscle mass, function, and bone mineral density in pre-sarcopenic and pre-osteopenic men and women aged 50 to 70.
Milk is rich in high-quality proteins. We in FHI believe we can provide the scientific evidence for the development of new products in the support of healthy ageing and performance nutrition.